Hi, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by two guests, Monika Vezse and Dr. Lee Williams. Listeners, Monika is the EMEA Market Segment and Application Lab Manager for Analytical Testing and Diagnostics at Biotage, and Lee is Director of R&D for Analytical Consumables and Applications at Biotage. And today we are going to talk more about Biotage and their work to support anti-doping efforts in Europe, the Middle East, and Africa. So Monika, Lee, welcome to the show today. How are you?
Thank you so much, Marie. We are, I think, both very excited to be here today with you.
Yeah. Hi, Marie. Doing good. Thank you. And yourself?
I'm doing quite well. Thank you for asking. And we're excited to have you both here with us today and to learn more about you and what you've been up to at Biotage. But to give our listeners a little bit of background here, can you start by telling us more about yourself and your role? And Lee, perhaps we'll start with you.
My name is Lee Williams. So I'm the R&D Director for Consumable Products and Application Development at Biotage. So I'm based out of Cardiff, which is our Wales site in the UK. My background is in analytical chemistry. So I did a PhD in hyphenated chromatography mass spectrometry techniques many years ago. So I kind of joined Biotage to head up the mass spec function.
Very cool. And Monika, how about yourself?
I'm Monika Vezse. I look after Biotage's Analytical Testing and Diagnostics segment across Europe, the Middle East, and Africa. My background is in also analytical chemistry, and I've been in this field for over 15 years, working across different regions on everything from lab automation, sample preparation. In my current role at Biotage, I focus on bridging the science and business. I work closely with the analytical laboratories, sales teams, and global partners across the region to develop new strategies, introducing technologies, and to make sure that our solutions meet the real-world needs of our customers. And a big part of my work is leading up our application lab in Cardiff, where we collaborate with laboratories on demonstrations, workflow optimizations, as well as knowledge sharing. So while my background is technical, my role is really about connecting people, solutions, and the science, which is the part that I enjoy the most.
Very cool. And I think scientific pathways are quite varied, particularly in the anti-doping field. I'd love to hear a little bit more about each of your pathways. So, Monika, how did you find your way to Biotage ultimately?
I actually started my career as a development scientist in the UK, working on chromatographic methods and later leading projects in food safety and regulatory affairs in collaboration with both the Chinese and the American regulatory authorities. And over time, I realized that I loved the science, but I also loved working directly with customers, understanding their challenges and translating solutions into practice. And that led me into more commercial and customer-facing roles, where I spent a lot of time traveling across Europe and later on in Asia, supporting customers on hand. Joining Biotage about 10 years ago now felt like a natural next step because it did bring together both sides of what I'm really passionate about, which is the strong science and the innovative technology, but on a much broader scale in this instance. So what really stood out for me was Biotage's focus on the real-world challenges in sample preparation and its collaborative culture. And that combination is why I still feel inspired to be here.
Oh, definitely. And Lee, can you share a little bit more of your story and how you found your way to Biotage?
As I mentioned, my background was chemistry with business studies back in the day. I took on a PhD then, which was analytical based on chromatography, mass spectrometry. So a lot of supercritical fluid and liquid chromatography, some GC during my PhD. After that, there really wasn't a lot of jobs in Wales. So I decided to open up my horizon and move to the US. So I signed up for a postdoc. I went to the FDA labs, National Center for Toxicology Research in Arkansas, of all places.
Oh, cool.
That was fantastic. I had a really steep learning curve, and that's really where I started my sample preparation journey. Obviously, as analytical chemists, we're all interested in the puzzle and solving the different puzzles, whether that be you integrating your chromatography, your mass spectrometry, or even the sample prep side of things. So it really grasped my interest. Towards the end of that, then I was looking for my next position. It was a toss-up between staying in the US or coming back to Wales, potentially in England. I saw a job pop up. The company was called Argonaut at the time. So, as I said, there wasn't a lot of jobs in Wales. So it was an opportunity to come home and work in the motherland. So that's what I ended up doing. So I came back in 2004 to join what ultimately goes on to be Biotage now.
Very cool. And you alluded to it just in that last sentence there, but can you share some of Biotage's history?
I've been around since 2004, so just over 21 years. This could be quite a long story, but I'll cut it short for the benefit of the viewers, or listeners, I should say. The company started off in the late 1960s, essentially out of a garage in South Wales. So Jones Chromatography popped up in 1969. That kind of went on. The whole Biotage portfolio was very different. So there were companies called Personal Chemistry, Pyrosequencing going on in Sweden. The Jones Chromatography is the part that myself and Monika have kind of grown through. So during the 90s, there was an extension from just chromatography phases, gas chromatography, GC columns, LC columns, and then a move into the sample preparation. So there's a merger with IST, which was International Solvent Technologies. Again, that was another spin out from, I believe, the University of Arizona. Mike Burke, who was a fantastic guy and mentor. That merger led into a lot of focus on looking at sample preparation, providing clean products to give the best analytical result. Through the 90s then, ultimately ended up in 2002, we were purchased by Argonaut Technologies, which were more of a flash purification company at the time. That lasted only a couple of years, and eventually Biotage purchased the majority of that from Argonaut Technologies. So there was a merger of the various parts: flash chromatography, sample preparation, some peptide synthesis and purification. And then over the course of the next 20 plus years, various mergers, acquisitions over the years lead us to what Biotage is today.
Definitely. It sounds like there have been a lot of changes and sort of this evolution over the years. So Lee, can you tell us a little bit more about what Biotage looks like today, and perhaps what kinds of products and services are in Biotage's product catalog, and what problems or needs do they solve in the market?
We're a global supplier. Our primary focus is separation and purification technologies. So business areas really focusing around drug discovery, development, but mostly analytical testing. So within our analytical testing portfolio, we cover environmental, a lot of clinical customers, forensic toxicology. So most areas, we've got sample prep and different workflows. So we are split up within the company. So there is an instrument and a consumable part. So we do look for full workflows associated with all of these areas. Sample preparation, everything from solid phase extraction to supported liquid extraction, protein precipitation, mainly around the small molecule environment, but we're focusing now towards the biological separations. Some of the other areas involve evaporation. So that's both in for peptide at post-flash purification and in the analytical segment. Automation, we got flash chromatography, purification systems, everything from small 5-gram columns up to multi-kilogram scale preparative columns. And I think over the last probably 6 years, a couple of most recent acquisitions have really started to push us in towards the bioseparations with different types of media and different size endpoint target molecules.
That's really interesting. And can you give our listeners some insight into, I guess, how anti-doping fits into the picture?
Yeah, we do a lot with both forensics and clinical tox. So I know the US market, there's a lot of work been put in with a lot of the big clinical houses as well as the smaller forensic units. I think anti-doping can be an extension of that. So certainly Monika has dealt with different WADA labs all over the continent, US based, pretty much everywhere across Europe and Middle East. Maybe not just in humans, but also animals. So we do a lot with different horse racing labs, camel racing, different doping areas.
Very cool. So let's talk about that footprint. Monika, can you give us some more detail about what Biotage's footprint looks like in Europe, the Middle East, and Africa, this EMEA region, and who do you serve in this region?
So Biotage has a very strong footprint across EMEA. We work with a wide variety of analytical laboratories, as Lee mentioned, everything from clinical and pharmaceutical companies to environmental and food testing labs, as well as forensic toxicology and, of course, the anti-doping labs. So most of the labs accredited by the World Anti-Doping Agency in the region are in regular contact with us and rely on our sample preparation solutions as part of their workflows. And beyond providing technology, we also collaborate closely with them through our R&D lab, Lee is heading up, and the application lab in Cardiff. And to me, what makes EMEA region unique is its diversity, and that really energizes me. And that's not only about the many languages and different cultures we are dealing with, though that's certainly a big part of it. We are also working with highly advanced centralized laboratories while also partnering with emerging labs that face very different resource and infrastructure challenges. And our job here is to support both ends of that spectrum, and not just with the technology, but also with training and method development and long-term partnership to make sure that they can really rely on consistent and efficient workflows, whatever their situation is.
Certainly. And it sounds like your products and services span a really broad range of different fields and kinds of laboratories. So can you maybe talk about what are the priorities right now for Biotage in the EMEA region?
One of our biggest priorities is really about future-proofing the labs. The challenges are always changing, of course, whether it's more complex substances, tighter regulations, or limited resources. So our focus is giving them a sustainable and scalable solution that help them stay ahead. And for anti-doping labs, that means making sure they have the tools and automation to keep up with the increasing testing demands and more sophisticated doping methods. And for other labs, it may be about efficiency or cost effectiveness or meeting strict regulatory standards. But across the board, our priority is to work side by side with these labs, like I said earlier, not only as a technology provider, but as a partner that really invests in their long-term success.
Certainly. And how do you balance, I suppose, the sort of keeping up with daily operations with this looking into the future and trying to anticipate what's coming down the pipeline?
So balancing the day-to-day activities, the immediate needs that are obviously driven by customer demands with the long-term strategies, of course, always a challenge, especially because we cover such a broad range of market segments like environmental, pharmaceutical, forensic toxicology, anti-doping, and so on. But this is exactly what makes the role engaging, and it really keeps us on our toes. I do see operations and strategy, however, not as competing priorities, but as connected, because we are constantly learning from the customers, their problems, their needs, from the regulatory bodies, from the conferences. And the feedback from the field is what helps us anticipate which applications and workflows will be critical in the future and how to prepare for that.
Absolutely. And Lee, is there anything you would like to add?
Yeah, I think it's really interesting because the company does have quite a wide portfolio. Certainly from my perspective, within R&D, we are looking at various areas daily. Obviously, there is a day job. If we've got product development or any ongoing studies, then that is scheduled in. But looking at where trends are going, hot topics, whether that be from an application standpoint or new kind of technologies that are starting to be adopted, we have a relatively good balance there. I think with the sample prep side of things, the sorbents that we deliver and make, it's not just for one particular area. So it could sample prep or an SPE media will go into environmental, it'll go into clinical forensics. So it might be a different format, it might be a different setup, different volumes, but ideally there's more than one coverage, if you like. So yeah, it's something that we do get pulled in different directions because other parts of the company has different focus. The environmental section will want X, Y, or Z. Clinical may have something. We are pulled around a little bit and have to support quite a lot of these different efforts.
Yeah, and I think it also helps that both in the application lab and in the R&D lab, we have a wide range of backgrounds. So we can rely on our colleagues to help and support us and our customers in different fields in that sense.
I think that is fantastic. It sounds like you have a wonderful team there, and you've been working with people all over the world to continue to advance their needs. So how do these customer needs maybe differ, Monika, across these different regions that you're serving globally? And perhaps could you share some examples of these different activities that Biotage has been involved in within the EMEA region?
In relation to the anti-doping world, the WADA standards equally apply to all accredited laboratories, and that framework provides an important global reference point for consistency. But regardless, the needs of the labs can still look very different depending on where they are. For example, highly centralized labs often focus on scaling up capacity, automation, and advanced workflows to handle high sample volumes, especially around major sporting events like the Olympic Games or any other international competitions. In contrast, smaller labs may be working with tighter resources, so our role there is to make sure they can still apply robust and reliable methods, even with the limited infrastructure they may have. So personally, I've been involved across this whole spectrum, from supporting high throughput labs during the international competitions to leading training programs and knowledge sharing workshops in regions where hands-on support is what was needed and makes the biggest difference. And that is the variety, what makes the work so enjoyable and rewarding, really, being able to adapt to the local realities while still working toward the same global mission and making sure that every lab can meet the WADA standards.
Definitely. Monika, I think it is so cool that you've had the opportunity to kind of be in the lab and help these labs make it through some of these large sporting events. I think there's a lot of pressure in a lot of these cases.
Absolutely.
And I know you worked with the French anti-doping laboratory, which is known as LADF, to prepare for the Paris 2024 Olympic and Paralympic Games last year. So can you just share a little bit more about this partnership and what that experience was like?
So we've been working with LADF for a long time, since 2015, actually. It started with introducing sample preparation techniques, and it's been very much a team effort with several of my colleagues involved over the years. And it has grown into a fully integrated, automated workflow. And what I loved the most was how efficient and professional the LADF team was. And they made it a real partnership. And it was just amazing to see how everything came together by last year for the Olympic Games. And there we didn't just provide the consumables and the instrumentation for the sample prep, of course. We were also working side by side with the LADF team to optimize their workflows for very high throughput and to deliver hands-on training ahead of the unique demands of the games. And it's not only that, but it's also important to mention that our service engineers were also on standby during the competition period, making sure that the systems run smoothly under the immense pressure of the Olympic testing. So we had spare parts in stock. They had everything handy in case something was to go wrong. But in the end, LADF successfully analyzed thousands of the urine and blood samples in just about 6 weeks under all the strict deadlines and protocols. And as you can imagine, we are all very, very proud to have played part in that achievement. And now looking ahead, we are excited to be preparing for the 2026 Winter Olympics, and this time together with the Italian WADA lab.
I think that is remarkable. So what was it like for you, Monika, being on the ground there in Paris, maybe either helping prepare or working through those games?
So during the games, I wasn't personally there. As you may imagine, there are strict rules to who can be present, and those people had to be vetted and had to be given special permissions. But the local account manager and the service engineers were there and always on standby. So that, unfortunately, that part I couldn't experience personally, but leading up to the games, I have also visited the lab several times together with my colleagues. So that's been a unique experience, even for me, even with the small involvement compared to those guys actually on the ground themselves.
Certainly. Well, Monika, it sounds like a phenomenal partnership and a wonderful experience to be part of the successful Olympic and Paralympic Games story there in Paris. And perhaps we could get into some of the details of what Biotage is doing and how you're collaborating in this partnership. So can you tell us a little bit more about the Biotage? We'll start with the Extrahera automated sample preparation workstations and the turbo evaporators that were implemented there at LADF. So Can you tell us just a little bit more in general about these instruments and what benefits that they provide?
So the extra-hera workstations and the TurboWeb evaporators were, like you said, key components to the LADF workflows during the games and, of course, before and after in their day-to-day activities. So the extra-hera is a compact automated sample preparation system that is designed for both speed and flexibility while remaining highly user-friendly. And this is the part that was crucial during the games because so many people had to use the system in a short time. So I forgot to mention actually that prior to the games, LADF underwent a major transition. They moved facilities to the Paris-Saclay University area, and they also doubled their workforce. And during the games, they have had many people from different WADA laboratories from all around the world joining them to help them out during the day-to-day testing. So you can imagine a user-friendly system was more than important in that sense. So they automate a wide range of techniques, like Lee mentioned earlier, including the supported liquid extraction, the solid phase extraction, the phospholipid depletion, the protein precipitation techniques using either plates or columns. The extra-hera actually comes in 3 different versions, depending on the sample volume itself. So the LV200, LV stands for low volume, handles very small volumes down to just a few microliters and is ideal for microelution workflows in 96 well plates. And then we have the HV, so the high volume 5000, which is built for high volume column-based preparation, handling up to 5 milliliters per pipette channel. And then we have the extra-hera Classic that gives you the flexibility to run either plates or columns. So it's really the best of both worlds. And then we have the TurboWeb family for evaporation after the analytes are extracted, depending on the sample format and the throughput needs. The TurboWeb 96 is all about high throughput with plates, which really help laboratories keep up with large sample volumes. And then the TurboWeb LV is focused on tubes and vials, handling up to 48 samples at once in volumes from 1.5 milliliters up to 60 milliliters. And then the TurboWeb extra-hera is all about the seamless workflow integration. So it takes tubes directly from the extra-hera systems without transfers. But that's just a quick overview, really. I don't want to sound too technical. But of course, I'm always happy to follow up if anyone would like to know more details outside of this episode.
Absolutely. And does the LADF laboratory have a mix of these different models of the equipment?
Yes, they do. Depending on the type of samples they analyze, whether it's blood or urine, they will require different volumes. So for that reason, they are using all of the different models that I just mentioned earlier, both for the extra-hera and for the TurboWeb evaporators.
Very cool. And I think beyond the extra-hera and the TurboWeb evaporators, there's additional equipment in the LADF laboratory from Biotage. So Lee, perhaps you could tell us a little bit more about the Biotage Pressure Plus manifold. What is this system and how does it work?
I think positive pressure from a column or a plate processing perspective has been around now for many years. It's superseded vacuum processing, which was kind of the original. So you'd literally have a vacuum processing unit that would pull either plates or column units in these manifolds. So positive pressure is a far more refined option. The difference between the two units, we have a pressure plus 48 system and a 96. Essentially, the 48 is more towards the column-based approach, whereas the 96 is for 96 fixed well plate formats. The advantage over conventional vacuum processing is that unlike having one drawer of vacuum, you've got individual pressure units supplied to each column or plate position. And that gives you far more uniformity across a plate. So it doesn't suffer from different flow rates, different hotspots like you traditionally get with vacuum processing. So it's a far more reproducible sample-to-sample processing for any of these biological samples. So yeah, definitely has changed the face of a lot of this processing.
Very cool. And in listening to you and Monika talk about the different products, it sounds like they work very well together kind of to make a streamlined workflow. Could you comment on that as well?
Yeah, so the workflow pretty much from taking your sample, reformatting it, and then from an extraction standpoint, you can automate the extraction with the extra-hera, or positive pressure if it's a semi-manual approach. That right through to the evaporation. Then when you've got your eluent from the system, you just pop that in the evaporator, that dries down, and then initially looking to resuspend prior to any LC or GCMS analysis.
Certainly. And I think a big change in the anti-doping field, I'm sure is in other fields as well, is this change from the larger tubes to the smaller 96 well plates for sample analysis. And this can boost efficiency and make it easier to do this high throughput processing. But there's then greater risk for potential cross-contamination or crosstalk, as some people call it, between the samples because these 96 well plates are smaller, they're closer together, and this is an issue for anti-doping. So Lee, can you talk about how Biotage products are maybe designed to help reduce this risk of crosstalk?
Yeah, absolutely. Look, cross-contamination or crosstalk, it's a concern whether you're dealing with columns or plates. Ultimately, it's the whole process from start to finish. We did a lot of work on a study, probably 5+ years ago, where we looked at the whole process from start to finish. Things like mixing can impact cross-contamination. Obviously, when you're dealing with columns versus plates, the spatial arrangement is something that is definitely a concern. Likewise with the evaporation. So we kind of evaluated all of the parameters involved from a volume perspective, mixing in a mixing plate. If you were dealing with 96 well plates, traditionally from a vacuum perspective, depending on the vacuum manifold manufacturer, there would be different gaskets. So the lure tips of the plates could extend to different lengths into the collection chambers. So the one thing that positive pressure units do kind of eliminate, likewise with the extrahera, is that from a crosstalk perspective, the eluent coming from whether it's column, lure tips or plates is always contained in the individual position so there isn't any potential for spluttering. Obviously, those units, we've put a lot of work into controlling the flow rates and the pressures that they're processed at. So there's guidelines for that we've come out with. Pipetting is a massive source of potential, especially if you're using a 96 handler or 8-position liquid handler where you potentially are moving over other sample wells. So what we focused on there is really looking at all of the pipetting parameters to provide a fully controlled pipetting option depending on your matrix, your viscosities, your solvent volatilities, etc. So you can change your flow rates. You can fully control pipetting heights, air gaps. And that's something that there was a lot of emphasis put on. Even the evaporation, the spatial design of some of these evaporators, you can get crosstalk even with column evaporation. So looking at the temperature control, can you run under cold conditions? There's potentially additives that you can add from a volatility perspective. Amphetamines are a case in point, where their volatility is such that they can cross-contaminate quite wide areas. Did an interesting poster at ASMS a few years ago to sort of detail this. But what we've concentrated on from the evaporation standpoint is really having good control of the temperatures. Some of the newer unit now, the plate format. So we've got programmable flows for gas usage, different temperature programs, individual channels where you can run one hot, one cold if required. So your adiabatic expansion of the gas can provide the cooling effects if needed, depending on your analyte volatility. So you really have fully tailored evaporation conditions. We've also actually included an elevator system. So you can move your plate closer to the nozzles where the gas flow is. So you can actually save on gas usage and evaporation time, which again is something that is extremely important for any of these assays. Crosstalk's an issue, but obviously evaporation is the bane of everybody's life. I did make a comment to senior staff when we were developing this unit. I've never been this excited over an evaporator system, and really is quite nice. So if anyone hasn't seen it, it's definitely worth a look.
I think that's phenomenal. It sounds like, Lee, from an R&D perspective, Biotage is not just developing entirely new products. You're also working on continuing to enhance the current products and services that you offer. Is that correct?
Yeah, definitely. You'll see across the industry, there's some moves, some drivers, but ideally highlighting application areas where existing products or existing workflows can fit quite nicely. So the last probably 5 or 10 years, we've really done a lot in forensics and clinical to really focus existing products to show advantages from a workflow perspective. And that's something that we continue to do now moving forward in sort of the peptide area. Oligonucleotides is something that we've put some large focus over the last couple of years. So yeah, it keeps us on our toes.
Absolutely. And it sounds like these partnerships with laboratories are key in terms of understanding what their needs are, what their challenges are, and how you can help them. So Lee, can you maybe comment on how Biotage works with laboratories to identify or maybe develop the best solutions to meet the needs that they're facing every day?
Something both myself and Monika have been heavily involved with over the years. I think first and foremost is really trying to understand what the customer is trying to achieve. What that could be, what type of instrumentation they have. Is it an LC? Is there a GCMS? Their competence. Some people are heavily involved in solid phase extraction, so maybe a different technique is not so widely accepted in some labs. So there are certain considerations that we have to make. And then from an educational perspective is trying to explain and show those workflow advantage or the benefits that a different approach would give. Quite often we've got more than one option to get the desired result. So I think it's really working with the customer to see what fits and what's best for what they want to achieve ultimately.
Absolutely. And in working with these customers, has anything that they've brought to your attention really surprised you?
A lot of things. I'm not sure how much I can actually say. Over the years, there's been multiple things I've seen in labs. I think everybody uses a standard pipette. I have been into labs where people use glass pipettes and are transferring liquids and not necessarily with a bulb. So that was quite an eye-opener. That was only maybe about 10 years ago. So I think health and safety has definitely improved since then. So that was just one entertaining thing I did see.
Certainly. And I think you can't know what the challenges people are facing or how people are even using your equipment until you get on the ground there and see it in action. So I love that this is something that Biotage prioritizes. And if our listeners out there want to learn more about Biotage or more about the work that the two of you do, Monika, what is the best way for our listeners to learn more?
The best place would be our website, biotage.com. And this is where you can find detailed information on our solutions, customer case studies, and all of our products and resources. And of course, I would like to encourage listeners to also follow Biotage on LinkedIn, where we share updates, insights, and stories from the field, really.
Fantastic. Well, listeners, definitely take a moment to get connected, follow Biotage on LinkedIn, and take a moment to check out their website and learn more about their products and services. And Monika and Lee, it's been so wonderful to have you here with us today and to learn more about you and more about the company. So thank you so much for joining us.
Thanks, Marie. Yeah, it's been a pleasure coming on, and thank you to all the listeners on the podcast.
Thank you so much for having us on today, Mary, and thanks to your listeners as well. It's been a pleasure to share our work at Biotage with you all. Thank you.
Well, it's been a pleasure for me to meet both of you and to learn more about everything that you're doing. Listeners, always wonderful to have you here with us as well. Please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and/or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.